Hi everyone, it is my pleasure to give you this brief presentation of our abstract entitled Clinical Whole Genome Sequencing Co-occurring Fibrillinopathies and Pharmacogenetic Profiling. As indicated by the title, in this project we set out to detect co-occurring mutations in the fibrillin-1 and fibrillin-2 genes. To this end, we analyzed our database containing roughly 550 genomes of patients with suspected Marfan syndrome or related disorders. Screening for disease-causing mutations from the approximately 3 million variants in every genome is similar to trying to find a needle in a haystack, but in this case, there were actually two needles. Let me jump straight to the results. Indeed, we detected 3 individuals from 2 families harboring co-occurring or digenic variants in fibrillin-1 and fibrillin-2, and some of which were already described previously as disease-causing in the literature. Interestingly, these digenic variants seem to exaggerate certain clinical features, such as aortic dilatation, which is only present in individuals harboring both mutations in both genes. As a first conclusion, we would like to emphasize that not only complex, but also aortic phenotypes may arise from multiple variants and are of high clinical relevance, in particular also for disease management and family planning. As a next step, as we already had whole genome data available, we used this data to generate pharmacogenomic profiles for the patients. We have already described this approach recently, which enables us to provide treatment recommendations according to international guidelines for currently 77 drugs. Indeed, one of the family members is a slow metabolizer of CYP2C9, which metabolizes losartan and could therefore potentially influence the dosage required for a successful treatment. We conclude that whole genome sequencing data may not only be used for diagnostics, but also for assessing the pharmacogenomic predisposition of the sequenced individuals. I would like to thank all the people involved in the project, as well as the funding agencies, and thank you for your attention.